We learned in earlier segments that enzymes dramatically increase the rates of chemical reactions, oftentimes by a million or billionfold or even more. So the question arises, with the bounty of enzymes that are present in cells, how are these reactions controlled? In other words, why is the cell not just a complete milieu of random reactions occurring at any given moment to use up all of the reactant and create all sorts of products, not necessarily in the rates that they're needed or at the times that they're needed? The way that enzymes are controlled is through a few different mechanisms. We will learn about one of those mechanisms here. That mechanism is referred to as allostery or allosteric enzymes. This is one of the ways that enzyme activity is controlled so that cells can produce substances at the precise time that they're needed in the precise amount that they're needed, so that the body is a closely regulated machine. Recall that in order for an enzyme to catalyze a reaction, the substrate or substrates, those are the reactants, must bind to the active site. This happens because the shape of the substrate is complementary to the shape of the active site. The active site then encourages chemical reaction by positioning the molecule perfectly to engage in reaction and forcing engagement between any reactants that are required to react. For example, in the case of a hydrolysis reaction, the enzyme forces water to come into close contact with the exact region of the molecule that is reacting. So what I've drawn out here and boxed in blue is the scenario where there is no allosteric regulation. In other words, whenever the substrate comes in and binds to the active site, a reaction is catalyzed and the product results. In most cases, there is need for the enzyme activity to be regulated based on various factors within the cell that dictate how much of the reaction products is actually needed at a given time so that the enzyme can provide that level of the reaction product. Allosteric regulation is one way that the body, or whatever organism we're working with, gains control over the activity of enzymes. To show allosteric regulation in our schematic, we need to modify it a bit. You will notice in the revised diagram, we've introduced a so-called allosteric site. An allosteric site is a site separate from the active site on the enzyme that binds a separate molecule. That molecule that binds here is not a molecule that actually reacts via catalysis by the enzyme, but instead, the molecule that binds here to the allosteric site results in a change in the shape of the active site to either promote the substrate and bind it to the active site and thereby increase the activity of the enzyme, or alternatively, the molecule can bind at the allosteric site and cause the active site shape to change in a way that decreases binding of the substrate to the active site and therefore decreases activity of the enzyme. When the binding of a regulator molecule at the allosteric site results in an increase in the rate of chemical reaction, an increase in the formation of products, we describe that molecule as a positive allosteric regulator. On the other hand, in the case shown in our illustration here, where the active site changed shape so that the substrates could no longer bind as well, we would describe that situation as a negative allosteric regulator. So a negative allosteric regulator is a situation where the active site shape changes in a way that decreases the production of reaction products. Now we should talk about feedback control. Feedback control we can think of as a special scenario of negative allosteric regulation. And this is a pretty clever way that a variety of organisms, including humans, have devised to allow them to control exactly how much of a metabolic end product is produced. In order to discuss feedback control, we need to first talk about metabolic pathways. Most molecules aren't created via one chemical reaction, but instead created via a series of chemical reactions that work one after another in series to make a variety of modifications to a starting material to create the final end product. We refer to that process as a biosynthetic pathway or as a metabolic pathway. In the case of the illustration here, what we see is that we start with a substrate, that is the starting reactant, binding to an enzyme. That enzyme in pink catalyzes a chemical reaction to yield an intermediate that we call number 1. And then these other arrows here represent additional chemical reactions catalyzed by other enzymes to ultimately yield a final end product. So in other words, it's not generally one enzyme that's required to make a complex metabolic product, but instead a series of enzymes that work via a step-by-step pathway. Much like you walk down a pathway to get from point A to point B, you don't just jump from point A to point B. The pathway is the way that molecules go from one structure to another sequentially to ultimately yield an end product that can sometimes be very different than the starting material. In an example of nature's molecular ingenuity, this end product, in many cases, acts to control the pathway as a whole. How does it go about doing that? Well, when an end product is created at sufficient concentration, meaning the cell has obtained all of that end product that it needs, at that concentration, that molecule can act as an inhibitor to one of the enzymes within the metabolic pathway. In other words, that particular molecule shown in purple here binds at an allosteric site. By binding at an allosteric site, it acts as an allosteric inhibitor by causing a change in the shape of the active site. Once that active site has changed shape, it can no longer bind to its specified substrate from the metabolic pathway, and thus that chemical reaction and all the ones that come after it in the pathway are shut down. Once the concentration of that end product in purple drops off enough within the cell, that end product will fall off of the allosteric site due to the low concentration of that molecule within the cell. Once it drops off of that allosteric site, falling away, then the enzyme, with the allosteric site now open and not occupied by the purple molecule, returns to the original form of the active site. So the active site can now go back and bind to a substrate, and the pathway can start again and run completely to the end product. Hence, we described this feedback control as a specific type of negative allosteric regulation because it is an example of a scenario where a molecule binds at an allosteric site, causing the shape of the active site to change in a way that decreases the activity of the enzyme. Hence, it is described as a negative regulator. So feedback control is the special example or special type of allosteric regulation where the final end product of a pathway controls the formation of further product by inhibiting one or more enzymes within that pathway. Let's look at an example of feedback control in action. Threonine, the amino acid shown here, can be converted through a series of 5 enzyme catalyzed reactions called E1 through E5 to yield isoleucine. This is an essential biochemical reaction because oftentimes when the cell is aiming to create protein, there's a shortage of isoleucine but an excess of threonine. And if that's the case, then it is useful for the cell to have this pathway to convert threonine into necessary isoleucine. However, at times, there can be excess isoleucine so that it is not necessary for more threonine to be converted into isoleucine. So how does the body regulate that? Well, in the case of this 5-enzyme pathway, the first enzyme is referred to threonine deaminase. That's E1 here. Enzyme 1 is threonine deaminase. And it turns out that isoleucine is an allosteric inhibitor of threonine deaminase. In other words, what happens is that in the normal scenario, threonine deaminase looks like this, with the active site here shown schematically with these triangles where the yellow substrate, that is threonine, docks into the enzyme and a reaction is catalyzed to take that one more step down the biosynthetic pathway. In cases where isoleucine is present at sufficient concentrations, isoleucine bonds at the allosteric site to act as a feedback control mechanism, because when isoleucine, the final product of the pathway, binds at this allosteric site, that forces the active site to change shape here and here so that the substrate threonine can no longer bind to it. And if the substrate threonine can't bind to it, then the reaction can't take place. In other words, as shown by this red X here, the reaction pathway is shut off at the start of the pipeline, threonine builds up in the cell, and does not get converted into isoleucine. At the point later on, when the concentration of isoleucine drops off in the cell, this isoleucine molecule, abbreviated as I in our green diagram in the upper right, will fall off of the enzyme. Once that falls off of the enzyme will return back to its original state here, where the substrate can then bind to the active site and the pathway can continue along to make more isoleucine as needed. This is one of literally thousands of known examples of feedback control where the final product of a pathway, such as isoleucine, acts to bind to an allosteric site toward the beginning of the pathway and ultimately shut the pathway down temporarily to control how much of a final product is produced.